Yes, so our next speaker, second to last, is Irina, and I wasn't even going to try to pronounce your last name, I'm sorry. Because I would do a terrible job of it. So Irina is the Chief Medical Officer and Head of R&D at Triplet Therapeutics. She's a neurologist and psychiatrist by training and has treated many patients with movement disorders, including HD. She's been involved in drug development with a focus on rare genetic neurodegenerative disorders since 2001. She served as medical director at EHDN 2009 and 10, and has remained committed to HD ever since. And she's excited about the recent discoveries of genetic modifiers for HD, and that Triplet team is now using to develop therapeutic targets for HD. So I'll hand it over to you, Irina.
Great, thank you very much. I hope you can hear me all right. I have to have the headset because we have a ton of planes flying over us today, so I needed to eliminate the noise. So first of all, thank you very much to the EHDN, but then particularly also to Ralph and you, Irina, for giving us an opportunity to present an update on our program. So I will be presenting data with our clinical candidate, TTX-3360, an antisense oligonucleotide that is targeting a genetic modifier, MSH3. It lowers MSH3 and halts somatic expansion, and the goal is to prevent onset and or progression of disease in both Huntington's disease, but also other repeat expansion disorders. So as part of today's presentation, I will be making forward-looking statements. This is the outline of my presentation. I will just go briefly over the concept of somatic instability because we have heard some good sessions yesterday about the concept, presenting some data on our clinical candidate, and ending with some clinical data from our ongoing natural history study. Now, somatic expansion can be seen as a second step in a two-step disease process. And we have heard this yesterday from Vincent Guillon. Repeat expansion disorders comprise a group of more than 50 disorders, and many of those disorders have been linked to somatic instability and somatic expansion, particularly to trigger the disease manifestation. And so while each disease comes with its own disease gene and with a specific sequence that is repeated and a significant length that needs to be reached in order to cause the disease, and therefore also different disease manifestations, ultimately there is this common second step across a whole range of those diseases, which is that the somatic expansion occurs. We know in HD it does not occur at birth, so it occurs sometime in life, and it is this somatic expansion that has been linked to the onset of clinical signs and symptoms, and it is this process that Triplet is trying to stop. And the next two slides just illustrate some of the key differences when treating a disease gene and when treating an upstream mechanism such as a genetic modifier to halt somatic expansion. Now, when targeting mutant huntingtin, this ever-increasing toxicity due to continued somatic expansion and ultimately the production of the aberrantly spliced exon 1 remains intact. So this whole ever-increasing toxicity is continuing, and we know from animal data that it's likely that about 75% lowering of mutant HTT is needed to actually see a therapeutic benefit. Obviously, this mechanism is only applicable to HD, so if we want to target another repeat expansion disorder, we need to develop a new AS or a new molecule against this other disease gene. We have generated our own data, and we have looked at published data and presented data, and intrathecally delivered antisense oligonucleotide that is not conjugated, does not reach the deep brain structures in sufficient concentrations to induce such a profound lowering of the target gene, while there can be actually quite high cortical exposure, so quite disproportional exposure in the different brain areas. And because of the critical role of HTT for brain development, there is a concern to intervene with a HTT lowering therapeutic too early in an individual. Now, targeting MSH3 as a genetic modifier that halts somatic expansion abrogates this ever-increasing toxicity, and we expect will also hold the aberrantly spliced exon 1 production, because it prevents a certain critical threshold of CAG repeats to be ever reached. Unlike for HTT lowering, we know that consistently data have shown that 50% lowering holds somatic expansion in different experimental models. And we have seen some data, quite intriguing data yesterday from Vanessa Wheeler. Because this is an upstream mechanism, it can potentially treat multiple, up to 30 CNS repeat expansion disorders. Because we don't see that intrathecally delivered ASOs can reach the deep brain structures, which are of course affected early on and profoundly in HD, we have chosen an intracerebroventricular route of injection of an ASO to really secure delivery to the deep brain structures as well as the cortex. And because MSH3 is not involved in any brain development or any other particular development, there is actually no concern going even after a very early stage or young patient population. Now, as we were looking at what target gene initially to select, so obviously we have selected MSH3, and there are good both efficacy and safety reasons. Obviously, MSH3 is one of the genetic modifiers that came out strongly in the GWAS dataset that Jim Guzella also just presented in April earlier this year. And MSH3 is particularly intriguing because it's linked to age of onset, but also progression using both the total motor score and also the single-digit modalities test. But when we look at the NOMAD database for healthy human subjects, and we look at the level of tolerance of loss of function, we can see that HTT and also the ataxin, so the disease genes themselves, have actually a highly or very little tolerance to loss of function, highly constrained genes, which of course raises concern by lowering them too much. The same is true for some of the other DNA damage response genes that came out as genetic modifiers. Now, MSH3 is only moderately constrained, which means that we have no real concern lowering it by 50% because there is tolerance to loss of function. This is also confirmed by knockout animals. So 2 different laboratories have created MS3 full knockouts, and there is no discernible phenotype. There's a normal lifespan and no higher increase in any tumors. Now, again, this is very different for some of the other DNA damage response genes. Even though there are also genetic modifiers, they seem less suitable as a therapeutic target. And of course, we do know that HTT knockout is embryonic lethal, again, pointing to a very different safety profile of lowering a genetic modifier gene. Another critical consideration when selecting a clinical candidate antisense oligonucleotide is the propensity to induce NFL. So this is a set of different ASOs that we have examined in rodents after ICV administration, and all of those ASOs target MSH3, but at different parts of the gene, so a different gene sequence. And what you can see here overall, that there is both a sequence, but also a dose-dependent increase in neurofilament light, and there are clear differences between the ASOs. And so for us, it was important to select a clinical candidate with very little propensity for an increase in NFL. Now, some more data on our clinical candidate TTX-3360. I've said to you before that we have done our own experiments looking at intrathecal as well as ICV administration. And when we do inject TTX-3360 intrathecally, we get in line with published literature, very faint distribution, very little distribution to these deep brain areas. Yes, we of course know, particularly in terms of somatic expansion, that those deep brain areas are really affected early on in the disease, showing somatic expansion. Now, when we inject ICV, you can see here, this is next to the lateral ventricle, and we see a very nice broad distribution throughout the deep brain structures, but then also in the cortex. And this is shown here on this slide. So this is a non-human primate study. We gave a single ICV injection of TTX-3360 in one lateral ventricle, so unilateral injections. And then we have sacrificed animals at different time points up to 12 weeks post a single injection at a well-tolerated, clinically relevant dose level. we are looking always at a normalization to 5 housekeeping genes for a robust normalization of the MSH3 mRNA. And what you can see here, that throughout the striatum and then also in the cortex, we see a sustained and significant knockdown. And our target is 50%, so to maintain 50% lowering. And we see that we maintain 50% lowering in most areas up to 12 weeks after a single well-tolerated injection, which of course encourages us to think of a very convenient clinical dosing strategy. And then as we looked into ICV compared to IT delivery, I mean, of course, the #1 criterion was that we do not deliver to the deep brain structures with an IT delivery. But we have also come to appreciate over the last year or so that this is actually not a particularly safe and convenient way to administer an ASO, particularly not if frequent IT administrations are needed. Now, when we inject ICV, obviously there is initially the need for an implantation, but we have lots of experience with this ICV device implantations. They have been used for more than 60 years, initially in neuro-oncology, but more recently also more and more neurodegenerative diseases. And what we can secure, as you have seen in the non-human primate data, that we do deliver to the deep brain areas, but we can also withdraw CSF, and thus this is very convenient for continued safety monitoring without actually having to do a lumbar puncture. We do know that once the device is implanted, repeat ICV dosing is quite easy to do as long as best practices are adhered to, that we are developing with our neurosurgeon consultants, and we take lessons learned from Irina that was approved for ICV delivery. So we envision that ICV delivery could also be done at home, so further making it more convenient to get the therapy for the patients. Now, I do want to end with some data from our natural history study, SHIELD HD, that we launched last year. And I do want to really express our gratitude to the patients and the investigators and all the site staff who were so great to really enroll the study ahead of time. We have enrolled 70 patients. We still have 68 in follow-up. So we are really grateful to have this data that will be so important as we design our phase 1 clinical trial. So the target patient population for our phase 1 clinical trial is pre-manifest and early manifest HD patients. And this is why we selected a similar population in our natural history study. So you can see here they were enrolled based on the CAP score into 3 different CAP score groups. They have a normal representation in terms of age and also the CAG repeat lengths. There's a roughly equal distribution between men and women. And what we have started to look at is the PIN-HD. The PIN-HD is a more refined criterion than the CAP score because it includes the TMS and the SDMT as clinical metrics to stage patients. And the PIN-HD has been suggested as an inclusion criterion for an intervention trial to enrich for informative patients. And so this is our plan to use a PIN-HD greater than 0 for our phase 1 clinical trial. And most of our patients in SHIELD HD meet this criterion already, but those who don't, we are confident that they will meet it over time because as the disease progresses, the PIN-HD increases. And we have had regulatory interactions that confirm that we can use the SHIELD HD study as a long run-in or as a placebo augmentation for our phase 1/2A proof of concept clinical trial. Now, this is data that were analyzed by Jeff Long. Again, very grateful to Jeff for helping us with these analysis. This is only over 16 weeks. So because of the rapid enrollment, we could look at the first data point, longitudinal data point. And of course, this is a very short time period for HD, but we are still happy to see that those clinical outcomes, the HD cap and then also the composite UHDRS, actually do show the expected small changes over a 16-week period. And we are, of course, delighted to be able to use this data to really design the best inclusion criteria, but then also also the best outcome parameters for our phase 1/2a clinical trial. And Jeff Long has already done some trial simulation informing us that using the coded volume as a primary outcome, we could have 80% power for 30% treatment effect with only 60 patients on active treatment over an 18-month period. So we will refine these analysis when we have actually 48-week data in early 2022 to then design the phase 1 study that we plan to start in mid-2022. And here you just see the NFL data, again, very much in line with the reported literature. And we were sort of just excited to see that there is actually a correlation between the baseline CSF, NFL, and the HD cap, again, telling us that if we can use NFL or even some of the biomarkers such as imaging, that this will give us an early read into the potential efficacy of TTX-3360. But importantly, the other aspect of SHIELD HD is to collect longitudinal large volume CSF samples. And again, we are grateful to the patients who are willing to do that, and our investigators who are collecting diligently those samples. We are using those samples to optimize our target engagement biomarker assay. This is an assay that is measuring MSH3 transcript in the RNA that is extracted from extracellular vesicles from the CSF. So we are collecting CSF ultimately from the lateral ventricles. We will be isolating the EVs and then isolating the RNA. And this is just a proof of principle where we have seen that when we treat cells in a dish with our ASO, we see, a marked knockdown. So you can see here, these are the cell cultures either treated for 24 hours with the ASO or 96 hours with the ASO. We can see a massive reduction in the MSH3 transcript. And we see that this is very nicely recapitulated in the exosomes, at the mRNA level in the exosomes. This gives us confidence that we can actually measure this in humans in vivo. And we will be doing non-human primate studies that will confirm that what we measure in the EV RNA is actually reflected in the deep brain tissue reduction. We can then also use the RNA, but this is a second priority, to measure somatic instability itself. And MeToo in our team has shown that the RNA and the DNA CAG repeats actually are very nicely correlated, meaning that we can use the RNA to infer DNA somatic instability. And with this, I come to an end. I have shown you that we are targeting the second step in a two-step disease process, a second step that is relevant for multiple repeat expansion disorders. We are starting our initial clinical trial, however, in HD. I've shown you that we will be using the ICV route of administration, procedure that has been used for many years. With this, we secure delivery to the deep brain areas as well as the cortex, and we are on track to do our CTA IND submission in the second half of 2022. And again, we are thankful to all the participants in SHIELD HD who have helped us generate this data and inform our phase 1 clinical trial. And overall, we are really grateful to many wonderful advisors, clinical scientists, patient advisors, and of course, a fantastic team at Triplet that is really working towards our phase 1 clinical trial. And I look forward to your questions later in the panel discussion.
So thank you, Irina. Very, very interesting results and expected outcomes. So I will now hand it over to Ralph, which is our last speaker. And afterwards, he'll tell you what we're going to do.